So this will be the last part of my three-part series on bacterial staining. In the first two parts, I talked about the acid-fast and also the gram stain. And if you haven't watched those videos, I would urge you to watch those first. And the links will be available in the description. And in this part, I will talk about the miscellaneous stains. So let's first look at the bacteria that don't take either gram or acid-fast stain. So firstly, we have the myco and urea plasma. So the problem with them is they don't have any peptidoglycan, so that means they don't have any cell wall. And as gram and acid-fast stain both are directed towards staining cell walls, myco and urea plasma won't be able to stain with gram or acid-fast stain. Next up, we have the spirochetes, Treponema and Leptospira. They are actually too thin, so we cannot visualize them because they are too thin. So even if they take gram stain or acid-fast stain, we can't see them like the normal bacteria. And lastly, we have the obligate intracellulars that are the rickettsial and chlamydial species. So they also don't take the gram stain. Actually, we have to stain both the host cell and the bacteria in case of rickettsia and chlamydia. So they also don't take gram or acid-fast stain. So first, let's discuss about Treponema pallidum. It's a very high-yield topic, and I will discuss two ways of visualizing Treponema pallidum. Firstly, we will discuss about the direct method. And why is it direct? Because we are directly visualizing the organism collected from the lesions that it causes. So the basic of dark field microscopy is the bacteria, Treponema pallidum, they actually don't take any stains. So they are unstained and white. So how can we visualize bacteria that are unstained and white? Well, we have to create a background that is in contrast with the white unstained bacteria. So we have created a dark background using the technology of dark field microscopy. So that's the basic behind this type of microscopy. Now, what are the advantages of this? Well, we can directly take the samples from exudates and lesions that Treponema pallidum causes. So we can take, let's say, fluids from syphilitic lesions and then prepare a slide and directly visualize Treponema pallidum in dark field microscopy. But there's a problem. The problem is it has a very low negative predictive value. So what does that mean? What it means is the test, if it comes out negative, we cannot say that we don't have the bacteria in the lesion. So that means we cannot be sure, even if we find a negative test, that the patient does not have syphilis or the patient does not have Treponema pallidum. Like in case of neurosyphilis, if we collect a CSF sample and prepare a slide and don't find any bacteria under the dark field microscope, we can say that there is a possibility that the patient is syphilis negative, but we cannot be sure at all. And one of the confirmatory tests is the Treponema pallidum fluorescent antibody stain, FTA-ABS, which stands for Fluorescent Treponemal Antibody Absorption. Well, I know it's a mouthful. That's why I have decided to explain it in detail, and that's why we will have to switch to the whiteboard. So let's do that. So to understand the FTA-ABS test properly, we need to look at the process in detail. But before that, well, what's the goal of the test? What are we trying to do? So actually, we're trying to detect this antibody-antigen complex. And this antigen is the Treponema pallidum antigen. And also, the antibody directed against that antigen would be the anti-Treponema pallidum. The anti-Treponema pallidum would be this antibody, and this is the antigen, and this complex is what we are trying to detect at the end of the test. So to do that, we will need, unfortunately, a number of antibodies and antigens. So we will have to know a few types of antibodies and antigens that are involved in this process. So firstly, we have a main type of antibody, and with that, we have a main type of antigen. And also, we have some unexpected antibodies that will interfere with the test. So to get rid of these antibodies, we will have to ensure that we have some extracted antigens. So those two will react and we will get rid of the unexpected antibodies. So now let's look at what exactly are the main type of antibodies and antigens. So the main type of antibody is taken from the serum of the patient, and the main type of antigen is taken rather from an animal source, not from a patient. And also we have to clarify that this serum obviously has to come from an infected patient. An infected syphilis patient has to give this serum to get the antibody. And the unexpected antibodies are directed against bacteria that are not Treponema pallidum. So these species are actually the other species of Treponema, but not the Pallidum species that is responsible for syphilis. And the extracted antigens, actually we get it from the lab, as I said. So this is all about the sources of the antibodies and antigens that are associated with FTA-ABS. So now that we are clear about the sources of these antibodies and antigens, let's look at what exactly are the main antibodies and antigens, and also the unexpected and extracted ones. So the main antibody is actually the anti-Treponema pallidum antibody. That's our main antibody that we get from the patient. And what's the main antigen? That's the Treponema pallidum bacteria itself. So we are dealing with the Treponema pallidum bacteria as the main antigen. And the unexpected antibodies, as I told you, are the anti-non-pallidum antibodies. And the extracted antigens are those bacteria that are non-Treponema pallidum. So non-Treponema pallidum bacteria are actually our extracted antigen. Now let's look at the steps that are associated with the process. We'll go sequentially. First off, we have a reaction between the unexpected antibodies and the laboratory extracted antigens. So that's our first step. So why is this the first step? Because we firstly want to get rid of the unexpected antibodies. We don't want these to interfere in our test, and this step is known as the ABS, or absorption step. So we are absorbing the unexpected antibodies. So this is the first step. And now the main reaction between the main type of antibody and the main type of antigen. So what will we get if we react these two? We will get an antibody-antigen complex. So if this is our main antibody and this is our Treponema pallidum bacterial antigen, this is the complex that we will get in step 2. So this is step 2. And in step 3, in the next step, we will actually try to detect this antibody-antigen complex. Now, to understand the last two steps a little better, I have included here a simplified illustration. So in step 1, we had some unexpected antibodies that could interfere with our test. So what we did was we added some extracted antigens, right? So then the unexpected antibodies with the laboratory extracted antigens, and then we got rid of this whole thing. We didn't have to think about the unexpected antibodies anymore. So with the unexpected antibodies out of the picture, now we have to deal with our expected or main antibodies and our Treponema antigen, Treponema pallidum antigen to be more clear. So if we get this reaction, as I said, we get these main type of antibodies from the serum of an infected patient. So if the patient is syphilis positive, the patient's serum will have these main type of antibodies, and we have provided these Treponema pallidum antigens from an animal source, right? So these animal sourced Treponema pallidum antigens will get to react with the main type of antibody if only the patient is infected with syphilis. That means the patient has anti-Treponema pallidum antibodies that are our main antibodies. So that's step 2. So from step 2, we got this antibody-antigen complex provided that the patient was infected with syphilis. Now, how do we detect? As I said, that our goal was to detect this antibody-antigen complex. So how do we detect this complex? Now we will have to introduce a new type of another type of antibody. So that would be our fluorescent antibody. So this, let's say, is the fluorescent label antibody, and this will help us detect the antibody-antigen complex that we have formed in step 2. So from step 2, we had this antibody-antigen complex. So this is our main antibody, and this is our main antigen. And now we have to detect this antibody-antigen complex. So now let's focus in this part, this antibody-antigen complex. So this is our complex, and we have this fluorescent label antibody. So how will we detect this complex? So as we can see that if we draw the fluorescent label antibody, we see that it fits properly with the Fc portion of our main antibody. So this is our fluorescent label antibody that's actually binding with the Fc portion of our main antibody, that's the anti-Treponema Pallidum antibody. Now this is our resultant complex. So this is our last complex that we were aiming for. We were aiming to get this ultimate complex, the complex containing the antigen, antibody, and also the fluorescent antibody. And how will we now visualize this? We will have to use a fluorescent microscope. So we will have to use a fluorescent microscope to visualize this fluorescent antibody labeled antibody-antigen complex. So that's the whole basis for the FTA-ABS test. That's the indirect treponemal test. So now that we're clear about the basics of FTA-ABS, we can look at the slide. So in this slide, we can see these are greenish organisms, and the green color is due to the fluorophore labeled antibody. And the main advantage of this test is that it is very specific for Treponema pallidum, because remember, in the absorption stage, we had absorbed all the bacterial antibodies against any species except for Treponema pallidum. So there is no chance that we will get a false positive result. And another advantage is that we can confirm a case of syphilis after we screen for syphilis with VDRL or RPR. So it can be used as a confirmatory test, unlike the dark field microscopy. And as I said, that the test is very specific for Treponema pallidum. So there is a very slim chance that the test will give false positive result. And also for its high negative predictive value, we can say that the test will also not give any false negative result. Because if we take a sample like a CSF sample for a patient of neurosyphilis, for a suspected patient, and if the test comes out negative, we can be almost 100% sure that the patient does not have neurosyphilis. So there are a lot of advantages for this test. Now moving on to the capsular stains, the principle that we should always remember is the capsule don't take any stain. So if we look at a few examples here, first, Streptococcus pneumoniae, this is a gram-positive cocci. So these are round-shaped, purple-colored gram-positive cocci here. And the specialty here is around the cocci, we see white clearing. Really dig deep, we can see that there is white clearing around clusters of bacteria. So these unstained areas around the cocci are actually the capsules, because remember, capsules don't take stain. And also, if we look at another encapsulated organism, that is Klebsiella, which is actually a gram-negative red-colored bacilli. Well, here also around the bacilli, we can see clearing. So this is also the capsule of Klebsiella. So that's something about gram staining and the capsules. But there is a specific test for capsules also, and that is the India ink stain. And we know that this stain is called the negative stain because we are actually not staining the organisms, rather we are staining the background. So the background here is stained with India ink stain, and the organisms here are not stained with India ink, rather they are unstained. And so we can say that the yeast Cryptococcus, they are unstained, and that's why we can say they are encapsulated. So that's about the capsules. Now another high yield stain, the periodic acid-Schiff stain. And the purpose of the stain is to visualize complex carbohydrate particles. And there's also a bit of chemistry associated with this stain. So to understand it better, we will switch to the whiteboard. So before we can start talking about the periodic acid-Schiff staining, there is a common mispronunciation that I'd like to address. Periodic acid, the formula for that is HiO4. So it's an acid of iodine, right? So if we try to explain a bit of chemistry, we can see that here we have a proton that's part of the periodic acid, and also we have an anion that is this one. So if we take a look at this, we can say that the oxidation state of iodine in this radical or anion is +7, and this is the highest oxidation state that can be achieved by iodine, right? So as in the periodic acid, the oxidation state for iodine is +7, which is the highest it can ever achieve. That's why we get the term per. So that's why we can say it's a anion or a periodic acid. And now that we know the nomenclature and also the pronunciation behind the term periodic acid, I think with a little bit of respect to chemistry, we should pronounce it periodic and not periodic. So what's the goal of periodic acid-Schiff staining? The goal here is to visualize the complex carbohydrate particles. So we want to visualize the carbohydrate particles, and more accurately, the complex carbohydrate ones, right? So we need to do actually 2 reactions to get this. So what's the first reaction? Let's look at the reaction here. So for the reaction, we need, as always, a reagent, and then we need a substrate. So we have a reagent and a substrate for the first reaction. So what's the reagent? The reagent is the periodic acid. So this is our reagent for the first reaction. And what's the substrate? The substrate is the carbohydrate particle or sugar, which for the purpose of this reaction we will write like this. So we have a bond between 2 carbons and also 2 hydroxyl groups. So that's our sugar, that's the substrate for the first reaction. Well, as I said, the periodic acid here, the oxidation state for iodine is +7. That means the periodic acid or the iodine cannot be further oxidized. So that's why this has to be a very strong oxidizer. Periodic acid is a very strong oxidizer, and it will oxidize the sugar. So how will it do it? Firstly, the oxidizer will break this bond, right? This bond will be broken first by the oxidizer, and then the hydroxyl groups will be oxidized to an oxidized carbonyl compound, and in this case we will get aldehyde group. So the hydroxyl groups are oxidized to aldehyde. So that's our first reaction. So now the second reaction. The second reaction, well, we will also need a reagent and we will need a substrate like before. So what will be the reagent of this reaction? The reagent for this will be the Schiff reagent, the Schiff part of the periodic acid-Schiff stain. And the substrate for this reaction will be the product of the last reaction, that is the aldehyde group. Now we will do a reaction between the two, and what will happen is we will get a compound or a product, right? This reaction will yield a compound that's actually a colored compound, and the color is purple. And this purple color is the reason why we will see the slides as purple also, right? So the purple colored compound will dye or color the slides purple as well. Now, to understand it a little bit more better, let's switch to our slides and take a look at a periodic acid-Schiff stain slide. So with the basic chemistry regarding periodic acid-Schiff now clear, we can move on to summarizing the stain. That is, acid actually oxidizes sugar and we get aldehyde residue. And the residues then react with the Schiff reagent and we get purple colored compounds and hence purple colored slides. So this is a slide of the liver of the hepatocytes in glycogen storage disease. So there is a lot of glycogen inside of the hepatocytes, and the glycogen molecules are actually polymers of glucose. So they are polysaccharides. And remember, what was the purpose of periodic acid-Schiff stain? The purpose was to visualize complex carbohydrate particles, and glycogen here is a complex carbohydrate. So here we can see a purple colored slide of liver in glycogen storage disease due to the periodic acid-Schiff staining. And as we are talking about bacterial stain, it will be incomplete if we don't talk about Whipple's disease. Well, Whipple's is a bacterial disease that is caused by the bacteria Treponema whippleyi. And here actually we are not visualizing the organism, rather we are looking at the lamina propria. So here in the lamina propria we see foamy macrophages. So that's also due to periodic acid-Schiff stain. Now our last two stains. Firstly, we have Giemsa, and this is used for rickettsia or chlamydia, the obligate intracellular. Here we can see that the rickettsial organisms are inside a host cell. And the last stain would be silver stain. Silver stain we can use for Pneumocystis jirovecii, which is actually a very important fungi in case of AIDS patient. And this is a slide of Pneumocystis jirovecii. So these black dots here, these are actually the fungi, and this is a silver stained slide. So that will be it for this video. And if we want to review the absolute high yields, we will obviously have to talk about Treponema pallidum, the two methods that I talked about in detail, the direct dark field microscopy and also the indirect FTA-ABS, that's the fluorescent microscopy. And these are very high yield. And also, last but not least, Cryptococcus, which is the causative organism for cryptococcal meningitis in AIDS patient. Here we can use India ink to visualize the characteristic capsules of the fungi. So these three are the main attractions of this video. And here I would like to end the video, and as always, keep learning and stay sharp.